Hi guys, my name is Rami Basbus and I'm from Harmony School of Science. Our project today is on enzyme catalysis. So to give you some background research, I'll be telling you what enzymes are and what they do. Enzymes are proteins produced by living cells, and their main purpose is to act like catalysts during chemical reactions. A catalyst affects the rate of a chemical reaction and in general speeds up the rate. One consequence of enzyme activity is that cells can carry complex chemicals at low temperatures, caused by speeding up of the reaction by enzymes. How this happens is the substrate basically binds to the reverse side of the enzyme, and the reaction is the slowing down of the substrate, causing complex chemicals to enter and be carried. The basic equation is enzyme plus substrate equals the enzyme substrate and the enzyme with the product. Essentially what happens in this chemical reaction is that the enzyme pairs with the substrate, and when it goes through the chemical reaction, the enzyme releases itself and becomes a product. However, the enzyme is not changed.
For our ELA component, we did journal entries that consist of 5 to 9 sentences for each week for 9 weeks. We wrote about our experiences of how we solved the mystery of enzyme catalysis. We also wrote an essay about industrial enzymes and how they're beneficial to the world.
For our social studies connection, we did a little bit on the history of enzymes. Enzymes were first discovered in 1833 by French chemist Anselm Payne. Later on, it was known that enzymes had a purpose, but never really did we know what they did. So, it went on to be worked upon by another French chemist named Louis Pasteur, who used the fermentation of enzymes with yeast and discovered ferments. So this led to basically enzymes becoming a major catalyst in biology.
Now to the important part of the project. We're actually going to be talking about the project. First, the materials.
Our materials are polyfoam pieces for various colors, pencil and paper, scissors, zip closed bag, tape, and glue.
Okay, so the procedure is to make a working model showing enzyme functions as a catalyst. Your model should contain an enzyme, which is the first color, this one, the substrate, colors 2 and 3, these two, and a competitive inhibitor, which is that color. Note the size of the polyfoam pieces you have received. Draw plans for your enzyme model or paper before you start to cut out your enzymes. They should be different sizes and colors so you can differentiate what they are. Do not make your model the same shape, as it says here. When all the components are drawn to scale, then you should begin cutting to make sure each and every one of them are equal in size and the same shape, color, etc. After you're done with all this, then you actually paste it on a piece of paper and label each and every enzyme product's competitive, non-competitive inhibitor, etc. When you're done with that, then you explain to your classmates what an enzyme does with its reactants, turning into a substrate and then releasing itself from the product, the end product. After you're done with that, you basically just talk with your classmates, as I've said, and then you tell everyone around you, your teacher, and you're done.
What he said.
Basically, so this visualization is of an enzyme substrate complex. As I've said before, the reactants bind to the enzyme and they go through the reaction, the enzyme substrate complex, and the enzyme comes out with the final product. The enzyme doesn't change at all, however. It could still be used further on for other reactions. This model is an enzyme inhibition, competitive and non-competitive visualization. Basically what happens is the same process, however there's a competitive inhibitor. And what happens is that competitive inhibitor binds to the active site and it interferes with the active site of the enzyme so substrate cannot bind. However, in the non-competitive inhibition, they change shape and still it binds to the enzyme but not at the active site, and the substrate still cannot bind.
Thanks for watching Harmony Public Schools.